Good morning, everyone. We'll get started here in a minute with our May Whiteboard Boardroom webinar. Our first presenters will be from the University of Missouri St. Louis. The Director of Technology Commercialization and Economic Development is Tamara Wilgers. The presenter will be Dr. Carl Bassi. He's a professor and director of research in the University of Missouri St. Louis College of Optometry. He'll be discussing the Q3D test, a handheld device to measure the amount of visual suppression and binocular function in patients. So welcome, Dr. Bassi, and anytime you're ready.
Thank you. Hello, everybody. This is a little bit different format for me. I'm actually not used to speaking on Zoom, so this will be kind of a new thing, but I'll try to just get going and kind of just relay the exciting things that we're doing here. Kind of the next slide, please. So the problem is, in terms of one of the things that's gone on in vision science is we've known about amblyopia for a long time. Amblyopia is sometimes referred to as lazy eye, and we've known about it, even the mechanisms of it, since the 60s. And despite our knowledge of it, despite it being a very treatable kind of condition, roughly, depending on whose numbers you look at, 2 to 3% of children in the United States still have amblyopia, which results in permanent vision loss. If you look at other areas in the world, those numbers are often much higher. So the question is what are we doing wrong? What are we doing wrong in terms of detecting things early enough? And one of the issues that's involved is one of the tools that's used for assessing amblyopia is to measure visual suppression. And I'll talk about what's the gold standard in just a couple of minutes, but the gold standard is not very good, and I'll explain why. What we've done is we've developed a much more sensitive test for detecting visual suppression. This started for me a long time ago. I used to work with patients with Alzheimer's disease, and one of the things that I was looking at in terms of visual changes in patients with Alzheimer's disease was these changes that happen in binocular vision, and I wanted to be able to screen for other kinds of binocular vision problems. And that introduced me to some of the problems in terms of diagnosing visual suppression. But I saw that the test was being done at the time clearly wasn't a very sensitive kind of measure. So can I have the next slide, please? So what was needed? I mean, I had the idea at the time of a way to kind of get around this. The old test is called the Worth 4-dot test. It's a qualitative measure. It only gives you sort of very rough kinds of information. And I wanted to come up with a way to have a quantitative measure of visual suppression. And one of the things that was lacking at the time when I was first thinking about this was just some of the light sources that were available really didn't give you a large enough range and intensity that's needed in order to be able to really do a good job of quantifying things. Now with the new technology, with new LEDs that are available, there's certainly the technologies there in terms of the light availability we need to be able to detect it. And the other thing that was needed was a way to really make sure we're separating visual input coming into the two eyes, and to be sure that's happening across a broad range of intensities. And again, kind of newer technologies using pulse width modulated control of intensity allows us to keep this constant kind of stimulus wavelength that's entering the two eyes. So myself and my two colleagues, Michael Howe and Wayne Garver here at the university, we developed a test that's very, very fast to administer. It's simple to do. It's a handheld device. It's inexpensive to produce, and we've already used it in a couple of clinical trials, a small trial that I'll describe in a bit, and a much larger trial at one of the pediatric hospitals here. So I will talk about that in just a minute. Can I have the next slide, please? If we want to know what the market is, there's a big market. This tool could be used by any number of vision care kinds of individuals, optometrists, ophthalmologists, technicians. One of the groups that's not on here, but certainly is a group that has an interest in this, is school nurses for doing vision screening. And because the Q3D is quantitative and more sensitive, it's going to allow us to detect problems earlier, which gives us a much better outcome in terms of treatments kinds of things that happen. The other thing is because it's quantitative, it gives us a tool to measure whether our treatments are actually working or not. If you have a quantitative measure, you can compare your baseline measure, you can see how things are happening now and see whether things are changing over time or not. And I think one of the other things that's really exciting about this is as we kind of learn more about other kinds of conditions, there's certainly other kinds of ways this could be applied, including assessment in things like optic neuritis and traumatic brain injury, where all of those have binocular issues that are associated with the two. Can I have the next slide, please? So what's out there? I referred to this already, the Worth 4 dot. I'm doing air quotes now. This technology that's being done it's nothing more than a flashlight, and this test has actually been around since the early 1900s in one form or another, and it really hasn't changed at all since the early 1900s. A person wears red-green glasses. They look at the flashlight and look for whether they see these dots that are on there. It's called the Worth 4 dot because there's 4 dots that are on there. And again, the idea is if you're suppressing an eye, you won't see that colored dot that's in the flashlight that's there. The problem with the Worth 4 dot test is it's really a blunt instrument. Some people have referred to it as the worthless 4 dot. It's only really useful if there's significant amount of suppression, and often that may be too late to prevent or reverse any kinds of things that are happening with the amblyopia. Have the next slide, please. Cameron, I think you were going to say something here.
Yeah, so I just wanted to let everyone know that we do have an issued patent in the US and also in Canada. And again, the claims cover the device, kits, including the device filters and methods as well. And I can certainly, answer questions offline or later in the presentation about the IP if you'd like. The patent issued several years ago, but it does still have a decent life on it. Okay, go ahead, Carl. Next slide.
So where are we with things? Well, we're really far along. We're actually on our 3rd generation prototype. Our original prototype was a plug-in model. It had a cord for it. Our 2nd generation, we modified it so we could use the device on the end of an ophthalmoscope handle, the standard kinds of handles that you see in most kinds of medical offices. And our 3rd generation that we developed with a University of Missouri Fast Track Award is a handheld device, and it's pictured there. It's obviously in a very form function, very ergonomic kind of design, and all the buttons are easily seen and can present the stimuli to the patients. We have 10 working prototypes. We have all the hardware, software, CAD design, circuit documentation already for this 3rd generation prototype. I mentioned we completed 2 clinical trials. The first trial was done at UMSL, and this was just to see kind of how reliable the test was, how repeatable the results were in an initial sample, both in patients with normal vision and in a handful of patients that had clinical conditions. The test was very reliable and very simple to run. It could be done, as I said, in less than a minute. We took our device over to Cardinal Glennon Hospital, part of St. Louis University, and worked with Oscar Cruz and Bradley Davitt. They're both pediatric ophthalmologists there at Cardinal Glennon, and we ran a much larger trial there with 350 consecutive patients. And what we did with those patients, we used our Q3D and compared it directly to the Worth 4-dot test. Just our findings was the Q3D was significantly better in terms of detecting issues, and was able to detect issues that were very, very small in terms of the amount of suppression that was present. So we have a working model. We have a validated model that's been shown to work in clinical populations. This is also run by the technicians over there, so it's something that doesn't require a lot of formal training in order to be able to operate. Can I have that last slide, please? So what's needed? Well, we're just about ready to go to market. We have patent protection, we've got our near production ready prototypes, and we've got clinical trial evaluation. What we really need now is a commercial partner to do there's just a few final design issues that we identified dealing with our battery, and it's not going to be a big issue. It's just the initial battery that we use, that initial battery is no longer available, so we're just going to have to come up with a new battery that will be used for it. We need to get FDA evaluation, but we've already had initial evaluations from a number of sources that says it will likely be an FDA 1, Class 1 exempt, and help with manufacturing, marketing, and sales. This is a great product. This is something that's going to really, I think, be part of the standard kit for people doing eye care in the future. And there's lots of other kinds of potential applications once people have this tool in their hands. I'll be glad to answer questions if anybody has any.
Yeah, can you hear me?
Yes.
Hey, Jim Waddella calling. Great presentation so far there. So question I had was, how does this, have you talked to anybody in the insurance world, obviously vision insurance, because it seems like that's the natural place where if you can do early detection and treatment, you're potentially saving saving treatment costs down the line.
We haven't talked directly with anybody like that, but one of the things we actually incorporated into our final prototype was what I didn't show on there was another light that could be used, and our thought was it actually be part of a separate billing code. If you do a sensory motor billing code, you can actually run both the sensory and motor part right directly from the device. So it should be used. It should be able to be used as a separate billing code just for the device itself.
Got it. Thank you.
Are there any other questions at this time? If not, Tamara, Carl, appreciate the presentation this morning, and if there are others out there that would like to exit or like to know more about this particular innovation, you can feel free to contact me directly. I'll get you back in contact with Tamara and Carl and see if we can move the technology along. So again, thanks again, Tamara and Carl.
Thank you very much.
Thank you.
Next up, we have University of Kansas Medical Center. The presenter will be Ashwini Betha. He is Senior Licensing Associate for Life Sciences at the Kansas University Center for Technology Commercialization. This particular technology, a couple of principal inventors, Peter Rowe, University of Kansas Medical Center nephrology professor. He's also a faculty member in the Kidney Institute and a full member of the University of Kansas Center for Drug Discovery, Delivery, and Experimental Therapeutics. The other principal inventor, Laird Forrest, professor in the University of Kansas School of Pharmacy and Pharmaceutical Chemistry. presentation will be on a novel hair growth enhancing formulation for the treatment of male pattern baldness. So Shwini, whenever you're ready, go ahead.
Thanks, Jim. Good morning, everyone, and thanks to Jim for giving us the opportunity to present one of our exciting technologies. This has been one of our featured technologies in our patent portfolio. Next slide, please. So as Jim just told us, the formulation is the subject matter here. The formulation for treatment of alopecia. So the main problem that we are trying to address here is this alopecia is affecting up to 70% of men and 40% of women. And American hair loss sufferers, they spend more than 3.5 billion a year for hair treatment. And at the time of 2010 and 2012, an estimated population of 310,000 undergone surgical hair transplantation. And right now, I was just looking at the market this morning. The total market for hair loss treatment all over the world has reached $5 billion in 2017. And US had the most hair restoration procedures performed. So the market size is huge here, and there is a need for a better hair loss treatment technology. Next slide, please. So how does our product solve this problem? Our product is actually a peptide discovered by Dr. Peter Rowe at KU Medical Center. And he collaborated with Dr. Lade Forrest at KU Lawrence campus to come up with a novel formulation based on the peptide that Dr. Rowe discovered. This invention has actually started when Dr. Rowe recognized a huge hair growth when he was working with animals in another project, and that actually triggered this hair growth invention. So right now we have a formulation. It's a topical application for treating baldness in both men and women. But the data we have right now is animal data. And we also have a focus on using this formulation for patients who undergo chemotherapy. As many of you know, hair loss is a big side effect for cancer patients undergoing chemotherapy. And what's interesting with our formulation is it's safe and painless, totally non-invasive, and it can be used in the form of a gel, just similar to products in the market like Rogaine. Next slide, please. Yeah, as I said, it's non-invasive, it's painless, and there's no need of hair transplantation, and there are no adverse side effects, even at high doses. It has the potential to replace the currently ineffective and painful treatments. Next slide, please. So minoxidil, minoxidil is a compound that is the active ingredient right now being used in several hair growth products such as Rogaine. And we have data in the animals showing that our formulation works better than minoxidil. I'm going to show the comparative data in my next slides. And our inventors, we have a co-inventor, Dr. Dan Aries, who is also the chair of dermatology at KU Medical Center. Dr. Aries believes that minoxidil may not work very well for most patients. 15% will experience some sort of hair regrowth, and 50% have hair loss delayed, and about 35% will continue to lose hair even after using hair growth products that are currently in the market. Next slide, please. In terms of intellectual property protection, we have solid 2 issued patents. The first patent is actually on the peptide itself. That led to our formulation development, and the second patent covers the peptide for the hair growth technology treatment. And we have other additional patents pending right now, and we have plans to file more patents as we come up with novel gel formulations. Next slide, please. So this is the stage of development. We have animal data right now. If you see on the left panel on the top side, Miracle is the name given by our inventors for our formulation. Miracle means a nice hair in Korean language. You can see the mice that recovered the hair growth after giving the formula, after injecting the peptide, in fact. And we do have data showing that our formulation is permeable and it penetrates the skin. The lower side picture on the left panel, it's the control, the vehicle that doesn't have peptide in it. And you can also see minoxidil growth on the right-hand side. And these have been quantitated on the right side panel B. So here what represents is the lower pixel density is proportional to more hair growth. So the lower the pixel density is, the more the hair growth is. You can see the first one is the control, no peptide, no formulation. The second one is a KU's Miracle formulation for hair growth. And the other control is the minoxidil that's currently being used for hair growth. So you can see that Miracle works much better than minoxidil in the animals. Next slide, please. So what do we need for further development? We want to collaborate with researchers and also seeking funding, either to start a new company or investors to invest on determining the long-term stability of the formulation, and also comprehensive testing of diverse gel formulations, because Dr. Lade Forrest is an excellent formulation chemist. He came up with several formulations based on this peptide, and we want to choose the best one eventually. So, and we still need to do testing in humans, and this is available for licensing at this moment. Next slide, please.
That's the final slide.
Okay, thank you. I'll be happy to answer any questions.
Shwini, what's the regulatory compliance situation? What would be involved going forward as far as classification and testing?
Well, as I said, Jim, at this point we have animal data only, and the regulatory issues will come up once we start doing human trials. This is not a medical device, so I don't think it's what is that, 510K or something, and right. I think this falls under either Class A or B. But I'm not a regulatory expert. Yeah, we don't have much of homework done on that side.
Okay, okay. If there are no other questions, again, feel free to reach out to me if you like to get in touch with the Shwini and or the inventors. Be happy to do that. Or if you have some questions you'd like us to pose back to Dr. Rowe or Laird Forrest, would be happy to do that as well. So thanks again for attending, and don't hesitate to reach out to us. Thank you very much.
Thank you.
Got it. Nice job.